Hi everybody. If you're watching this video, hopefully you watched the one from yesterday's lesson on Mendelian inheritance patterns. Today we're going to build off of that into more complex inheritance patterns. You can find this in your textbook. It's topic 1.2, concepts 3 to 5. So yesterday we talked about traits that have a dominant allele and a recessive allele, and these are called Mendelian traits. Most traits are actually non-Mendelian though, and are controlled in more complicated ways. So for example, traits can have incomplete dominance. That's when neither allele is completely dominant, so the heterozygote, the one with one of each type of allele, actually show an intermediate or a middle phenotype. So for example, this happens in a lot of flowers. When a red carnation and a white carnation are crossed, the resulting offspring are pink. So here is your red carnation, and you can see here that they have used capital letters for both the red and the white. They used C for color, and then they have an R for red and a W for white. That is because neither one of these are dominant to the other one, so it didn't make sense to have, for example, a capital C and a lowercase c because one is not recessive. You can also just use two different capital letters, something like R for red, or R times two W's for white, and then your heterozygote would look like this. So the intermediate between red and white is pink. So carnations can come in red, white, or pink flowers. So if you cross two pink carnations, this is what your Punnett square would look like. You'd have a red carnation allele from one parent and a white from the other, from the same parent. And then the other parent would be the same, one red allele, one white allele. And you're going to cross them just like you did in the Punnett squares from yesterday. And you end up having this cool pattern of having one of each of the two parent phenotype, or the red phenotype and the white phenotype, and then two of the intermediate or pink phenotypes. Let's try a practice question. There's a breed of dog that has hair texture that is determined by incompletely dominant traits. The hair texture can be straight, so I used T for texture and S for straight. Wavy, this is the heterozygote, has one of each, or curly, so two C's. If a straight-haired dog is crossed with a wavy-haired dog, what is the probability the puppies would have wavy hair? And what is the probability that the puppies would have straight hair? So I'm going to get you to pause the video here and give this question a try, and then hit play when you are ready to see the answer. Okay, are you ready? So you're making your Punnett square. And our question asked, what if you crossed a wavy-haired dog and a straight-haired dog? So our straight-haired dog is like this, TS, TS. And our wavy-haired dog is our intermediate phenotype, TS, TC. Okay, so now we're looking at our offspring. We have TS, TS, and then the bottom ones are both TS, TC, TS, TC. Okay, so it says, what is the probability the puppies would have wavy hair? So let's write a genotype and a phenotype ratio. So here we have 2 TS to 2 TS TC, and ratios are just like fractions, so if you can reduce it by dividing the 2's by 1 TS, 1 TS-TC, that is even better. So this means that half of the offspring are straight hair. Sorry about that. So they're equal numbers, straight and curly. I actually prefer to use percentages to show. So for example, I would prefer to say 50% straight hair and, oh, sorry, this is not curly. My bad, this is wavy. So 1 straight to 1 wavy. I hope you were shouting at your computer, Miss Fulton, that's wrong. Okay, there we go. Cool. So the question, always good to check the last sentence of the question. What is the probability the puppies would have wavy hair? What would be the probability of them straight hair? So that's a 50-50% probability, 50% probability of each. Awesome. How did you do? So that is called incomplete dominance. You can also have traits that are determined by codominance. Co means together, like cooperate, right? So codominance is when both alleles are expressed in the heterozygote. So for example, in cows, you can have a white-haired cow. You can have this red type of cow. They're called roan. It's like a reddy brown. And then you can have this cow that has spots. It's white with brown or red spots. Okay, so this is codominant. So it's important to the distinction between incomplete dominance and codominance. If you cross a red and white flower and you get pink, you get some new phenotype that's in between your parent phenotypes. That's incomplete dominance. But if you get white and red spots, that is codominance. If you see both phenotypes of the parents in the heterozygous offspring, that is codominance. Okay, so that's the difference. One looks like the middle ground between them, between the two parents, and one shows both phenotypes from the parents. So another example where this happens is in hens. So let's practice. It says, make a Punnett square showing the possible genotypes of crossing a speckled hen and a white hen and give the probabilities of each phenotype. So here we have, in hens, there's a codominant pattern. You can have a white hen, you can have a black hen, I should say chicken, white chicken, black chicken, or a speckled chicken, okay? White we're gonna call big W, black, big B, and speckled is our heterozygote, so 1B, 1W. So in our Punnett square, we're showing a speckled hen and a white hen. So draw your Punnett square. Oh, pause here. Try it yourself. Pause here so you can see the question. Try it yourself and then come back when you're ready to move on. Welcome back. I'm going to show you my solution and you can check your answer. So we have a white hen and a speckled hen. Yes. So the white hen is W. It gives all W alleles, all white alleles. Speckled hen gives 50% black alleles, 50% white alleles. And then we fill in our table, BW, WW, WW. Okay. So now the question asks the probabilities of each phenotype. So what phenotypes do we have here? Well, BW is speckled. And 2 big W's is white. And it wants the probabilities. So you can write this a couple ways. You can write a ratio. So you could do 1 speckled to 1 white. You could do a fraction. So this would be 2 out of 4. Reduce that, 1 out of 2. It's the same for white, of course. Or like I said before, I like to work in percentages. So 50% speckled, 50% white. So I hope you got 50% and 50% again. Give yourself a high five if you did. Okay, so far we've only talked about traits with 2 alleles, but actually many traits have many different alleles. This is called polygenic inheritance. We're going to start with a simple example of this, and that's blood type. So there's many different alleles, but an individual can only have up to 2 of the alleles, 1 on each homologous chromosome, or as we've been saying, 1 from mom, 1 from dad. So for human blood types, that's an example of having multiple alleles. There's 4 phenotypes. You can have blood type A, blood type B, AB, or O. And there's pluses and minuses too, which we'll talk about in a second. So geneticists usually do this. They call the gene I for blood type, and then they'll do a subscript like a little a, or sorry, superscript like an exponent, like a little a for type A. And the allele determines which antigens are attached to your blood cells, and antigens cause an immune response when your body doesn't recognize them. So for example, I have type B blood, so I have these little guys that stick out on my blood that say B, and my immune system knows that if it has these little flags that say B, then it's okay. That belongs to my body. If I needed a blood transfusion and they gave me someone else's blood, and this wouldn't happen because they do blood typing at the hospital, but say for some reason they've given me type A blood, my body would see this type A blood and it would say, wait a second, we don't have A's in here, this is an intruder, and they would actually attack it, and that would make me very sick. So if you have AB blood, you have both of the little flags, you'd have A and B. If you have type O blood, you have no antigens on your body. That's why type O blood can be given to anyone, because there aren't any of these little flags saying I'm type A or type B. And if you're more interested in blood type, I'm going to post a link to a really good video in the OneNote page for this assignment, and you can have a look at it and see a little bit more about why blood type is important. Oh, there's the video there. But I'm going to put it in your OneNote. So there's 3 alleles. There's capital I with an A for type A, there's capital I with a B for type B, and there's a little I. And little I is for type O. Okay, blood type O. And little I is recessive to big IA and big IB. A and B are codominant to each other. So if you have one A, one B allele, you have both A and B antigens in your type AB. Okay, so here are the different possibilities for genotypes. If you have 2 copies of A, you're type A. If you have an A and an O, you're A. So 2 B's, you're B. A B and an O, you're B. An A and a B, you're AB. And 2 little I's, you're O. Okay, so let's see. We might have to come back to this chart. So what would the genotype and phenotype ratios be with for a child with an AB mother and an O father? So pause the video here, give it a try, and come back when you need a hint or when you're ready to check your answer. Okay, welcome back. Let's do our Punnett square. You're gonna get so tired of drawing all these grids. Okay, so AB mother. So they have IA, IB, and an O father. So because their father is O and that's recessive, they must have 2 copies of the recessive allele. Okay, so now let's fill in our offspring. A little I, A, oh, yep, little I, B, little I. Okay, what would the genotype and phenotype ratios be? So we have 2 A, I, to IB, little I. And like I said before, we need to reduce when we have a ratio just as if it were a fraction. Okay, and then what phenotypes are these? Let's just copy down, but this is type A. Okay, we can go back to look. So an big A, little I is type A. And then it's the same with big B, little I is 1B. Okay, so hopefully you got 1-to-1 or 50%, 50%, totally fine. One out of two. Okay, I said we'd come back to talking about when your blood type is plus or minus. So that is because of something called the Rh factor. And the Rh factor, it shows whether your blood has these little D antibodies or not. So I said I'm type B, I have B antibodies. I'm actually B positive, also a good life motto. So I have B and D antigens. And Rh positive is the dominant phenotype here. So we could write this as big D, or big D, little d for that D antibody. So if you're positive, you have the D antibodies. If you're negative, you do not. So you have 2 copies of that recessive allele. So here's one more question for you to try. A child with a blood type of O positive has a father with a blood type of B negative. What must the genotype be for the child and father? And what are the possible genotypes and phenotypes for the mother? And this is a question that could actually be done in a hospital setting, because if the mother does not have the D antibody, she's Rh negative, and the child in her body has, when she's pregnant, has the D antibodies, her body might attack that fetus. So this is an important question. So pause it here, see if you can figure it out. A hint is that you are going to need 2 Punnett squares. Okay? Come back when you need a hint or when you're ready to check your answer. Okay, welcome back. So I gave you a hint that you need 2 Punnett squares. We're going to need 1 Punnett square for O versus B. Oh, sorry. So the child having O and the father having B. And then we're going to actually need a second Punnett square for the child having plus and the father having minus. So let's get to work. So let's first do the child of O has a father with a blood type B. So I don't think we've done these ones before where we're trying to work backwards. So if the father has a blood type of B and the child has O, we know the child's phenotype must be 2 little i's, okay? Because that's the only genotype that matches with the O phenotype. Now, if the child has a little I from dad, that means that dad's genotype has to have a little I in it, because this child has a little I from their dad. So then the other one is big I with a B, because they're type B. Okay? So now we're trying to figure out the possible genotypes and phenotypes for the mother. So that one I highlighted in pink came from dad. This one I'm highlighting in green must come from mom. So mom could actually be anything with an I. So she could be I A, I B, little I, or she could be type O herself. Okay, so you didn't actually need a Punnett square for that one, just logic. Okay, so those are her possible genotypes. What about her phenotypes? So she would be here, A, B, or O. Okay, now we need to know though for plus and minus. So, this child has a blood type of O positive, and because positive is dominant, we don't know what the second allele is. We just know that it has one copy of the dominant one. But the father has a blood type of B negative, so father, his Rh factor gene is 2 little d's because he's the recessive phenotype. So now actually we can tell that the child's phenotype, the child has to get 1 little d from dad. Okay, because dad can't give any big D alleles because he doesn't have any. Okay, so our child's genotype is I, and then heterozygous for Rh. Our father's is big B, little I, and 2 recessive, homozygous recessive for the Rh factor. So working backwards, let's see what the mom has to be. So let's go back to our Punnett square that I drew on the next page. So for mom, so we know dad is little d, and we also know that the child got a big D from somewhere. So mom has to be at least 1 big D. We don't know about this one. Okay, this one could be a big D or a little d. So mom could be, she could have the genotypes big D, little d, or big D, big D. Okay, and then phenotypes would be, she could be either, oh no, these both would be what phenotype? Both would be Rh positive. So mom is going to either be A positive, B positive, or O positive. Okay, you're going to go try some practice in your workbook now. Remember, if you have any questions, you can rewatch this video, you can call a friend, you can put a question in the Teams. That's a really good thing to do because someone else might have the same question and you're helping them out. And you can also email me. Let me know how it goes. Thank you very much, and I will see you tomorrow. Bye-bye.